Here we have, you are going to meet the women who are coding the future. There are three excellent professionals in this session one. And then we have speaker, dear friend. In December 2010, after a long stint as head of ophthalmic pathology lab at the Sudhakar and Srikant Ravi Stem Cell Biology Laboratory at the renowned LV Prasad Eye Institute. She holds several honors positions, including adjunct professorship of the ophthalmology in the School of Medicine and Dentistry, University of Rochester, USA. She also joined as a chair and program of the AP section of the Association for Research in Vision and Ophthalmology, and president of the Indian Society of Ophthalmic Pathology, Indian Society for Ophthalmic Pathology, and other professional and academic affiliations. Dr. Geeta Vemaganti earned her basic medical degree at the JLN Medical College, Ajmer, before completing her MD in pathology at Nizam's Institute of Medical Sciences. She has advanced training in ocular pathology and at Eye Institute, Los Angeles, USA, and has been visiting pathologist at the Eye and Ear Infirmary and Department of Pathology, University of Illinois. She has published more than 300 papers in national and international journals. She is recipient of several international and national awards, including Chemtech Foundation Award, Senior Achievement Award by American Academy of Ophthalmology, and also National Science Award for Career Development by DBT. So Dr. Vemagunti has received many prestigious academic as well as research grants from the national and international agencies and has guided many doctoral students in both basic and applied sciences. So with this, I'm very happy to invite Geeta Vemagunti to present her talk. Over to you, Geeta.
Thank you, madam. Thank you very much for your kind introduction. Is my screen visible? Am I audible and is my screen visible?
Yes, ma'am.
Yes, madam. Screen is visible, ma'am.
Okay. Thank you very much, Dr. Kaisar, Dr. Alka, Dr. Uma, and the entire team of this seminar. And I congratulate you for conducting this despite all the challenges. And this is, the topic is something which is very close to my heart, and that is research for its potential for translational research. And since the audience are from mostly engineering background and other mixed backgrounds, so I would just go through the basics so that we are all on the same page when we, so that we can understand the subject better. So why do we look at the stem cells so keenly, and why are we all interested in doing research in this? The reason is the human body is a fascinating machine, which is one of the most efficient machines. And I personally believe that the efficiency of this machine has a lot to do with its capacity to proliferate. So we can look at the body in either by anatomy, special different systems, but irrespective of all the systems, the way they develop is from one cell, it has become a full human body. So this is by two processes. One is by differentiation and one is by proliferation. And by a very well orchestrated developmental biology, one single cell becomes an entire human body. And that is one of the very fascinating things that we need to learn from. But if you look at the whole body, to simplify it, especially because of many of you are from engineering background, look at it like a combination of cells and extracellular matrices. So the basic component of the body is a cell. A group of cells is called a tissue. A group of tissues is called an organ. And different kinds of organ make a system. Now, when we talk about translational research, which part of which area do we want to use this stem cell research for. It's basically for regeneration or repair of these tissues. So that's the reason why we need to understand how the tissues function. So another way of dividing all the cells in the body, irrespective of whether it's a central nervous system or a skeletal muscle system or a cardiovascular system, we can divide all types of cells in our body into three types. One is a labile cell, stable cell, or a permanent cell. Why do we do this? This is because the repair capacity of our body depends on this proliferating capacity of the tissues. So we don't have to do a bone marrow transplantation if we get anemia or WBC counts go down. That's because the bone marrow regenerates itself unless there is some major problem. The other cells are stable cells or the SOS cells when they come to the rescue as and when required. The permanent cells we believe that don't have cannot repair, like central nervous system, skeletal muscle system, glomerulus, cardiac muscle, and all this proliferating capacity is we now attribute it to the presence of stem cells in these population of cells. So labile cells are easily repaired. Stable cells do have a repair capacity to a large extent. Permanent cells are the ones which have no regenerating capacity or are very limited, which does not rescue or come to the rescue if they are damaged. And all this proliferation is because of the presence of the stem cells. So how do we define these stem cells? These stem cells are the undifferentiated cells which have two distinct properties, which are capable of self-renewal and also differentiation. And this, if you recall the cell cycle, most of the cells are divided, labile cells are in the cell cycle. Permanent cells are in G0, whereas the stable cells are in G0, but they come to G1 and the entire cell cycle as and when required. So the challenge lies in identifying where are the stem cells and how do we said that from one cell an entire human body is repaired. So in that hierarchy, what are the stem cells? So in this one single embryo also, so from here to here, we have different kinds of stem cells. Embryonic stem cells, which are, they can be totipotent, pluripotent. So as you come down the hierarchy, the differentiation capacity becomes limited from generating the entire body to single organ. Like if you take the adult stem cells, all the labile cells and the stable cells we talked about in different parts of the body have the stem cells, but they cannot make different kinds of tissue like the embryonic stem cell does. It has a limited capacity, so therefore it will become, liver becomes a liver cell, skin becomes a skin cell, limbal becomes a corneal cell. It cannot become cardiac or a muscle. So that's the importance of the limited differentiation. So this is called multipotent, totipotent, pluripotent, multipotent, bipotent, or unipotent, based on the capacity to differentiate into different kinds of cells. So just because it is dividing, it is not a stem cell, unlike a progenitor. So the importance is the first characteristic, that is the self-renewal. So this self-renewal is both intrinsic as well as extrinsic property and is dictated by the niche as well. The unique properties of these stem cells, as I mentioned, can be intrinsic properties and extrinsic properties. And another key factor for this is it is an asymmetric cell division where one cell, daughter cell, goes back to self-renewal and the other one is pushed into differentiation. And we are all healthy, free of tumors, is because these are error-free proliferations. They don't divide with errors. And it's a quiescent, it's an underbunker area. Niche is something which protects these stem cells. So where are these adult stem cells located? We just talked about all the labile and stable tissues like skin. Skin has stem cells that repopulate the epidermis. It has stem cells which repopulate the sebaceous, some melanocytes. These are called bulk stem cells. Similarly, the crypts of the stomach, intestine have epithelial cells. All the skin and mucosa have the stem cells at the basal layer. And the mammary glands, all exocrine glands. And so, okay, we know about stem cells. So what are the translation? What are the potential areas where they can be further researched and put to application? So one of the most promising things everyone looks at is, can we replace the damaged tissues of the body by these stem cells? If we know adequately about these stem cells, can we replace the damaged tissues? That's the most attractive thing for any researcher. You can also have a disease modeling. If it's not just the, like we talked about cells, tissues, organ. So what do you want to make of from these stem cells? Do you want to make a tissue, an organ, so that you can study it as a disease model? Do you want a tissue engineering of a liver or a skin? Do you want to put gene therapy or genetic manipulation of these stem cells? And or so I will share my experience of going from basic research to translation in area of ophthalmology before we start with some of the advances in rest of the areas. And another exciting area of translational research is the targeting the cancer stem cells, which we started understanding better. So for replacement, uh, it's a challenge. Where do we get the cells from? So conceptually, if you take the cells from the same patient for the same type, like skin to skin, one part of the skin to another part of the body of the skin, it's autologous and homologous. So if it is same person, mesenchymal stem cells, but for another application of, say, peripheral vascular disease or for liver, it's called non-homogeneous application. Allogenic is some other person's um, uh, stem cell component to another, so it's allogenic, so it requires immune, or embryonic or induced pluripotent. These are the potential conceptual divisions, and you can see where which area would one like to work on. And so this is something that I'm going to talk about in my talk. How do we, which organs to make, what do we need to learn? What are the materials we require? What are the regulatory approvals? How do we understand the structure function relationship and how we translate it? So this is similar to all the 6 A's principle. First, you acquire knowledge, you assess your knowledge, you analyze, you apply, and you use the audit principles for any engineering purposes. So from all this in translational area, so which ones are the most attractive? So easiest ones are the epithelial and the bipotential cells, but the neurons are the permanent ones, which actually where we require stem cell therapy, that is the most difficult thing to proliferate. So let's see if we know enough about culture techniques by explant or by suspension cultures. If you know about the stem cells, are we ready to go for tissue or cell replacement or for tissue engineering if you want to use scaffolds? What is the prerequisite? So prerequisite is the regulatory approvals. The first thing is you have to have one slide later on regulatory approvals. You have to have a sufficient number of cells. You have to make, so you remember the two definitions, self-renewal and differentiation. So it should self-renew appropriately and it should differentiate appropriately into your desired cell type. And if you're going to clinical phase, then it is it surviving in the recipient? Is it networking, integrating, not causing any harm, or is it functioning? So this is a checklist that one needs to go before one goes for clinical. And it starts with the approval, regulatory approval. So in the hierarchy of these stem cells, which are the ones are we going to work on? Which are the ones we are going to use? We can also use established cell lines to do your basic research. So the questions is, when you're working on a particular type of cells, again, which source? Allogenic, autologous. Do you want to make the same cell? Do you want to make another type of cell? Do you want to use a differentiate? So this is a huge list of checklists that we have before we even say that we can go for therapy. So therapy is a very, very far-fetched word. It's just research is, that's why I'm saying translation research is what we need to talk about. And if you want to go for tissue engineering, then in addition to the cells, you need some molecules and you need some scaffolds to go for like a 3D picture. And what is the regulatory approval that you need to look at? If you're working on human samples, an institutional review board, which consists both of a scientific committee and an ethics committee should approve your proposal. If you are looking at stem cells of any source, in addition to IEC or IRB, you have to have clearance of institutional committee for stem cell research. And this is called ICSER. And if it's for any clinical trial, you have to go for NAC committee or the apex committee. So if you're using any samples for scaffolds, then you have to do the typical biocompatibility testing spectrum of all the toxicity testing. And if it is any engineering you have to do, then it is institutional